afternoon, everyone. My name is Jacqueline Zecca. I'm an application scientist here at OPS Diagnostics. I'd like to thank you for attending our webinar on a primer on microbial freeze drying. This webinar will be presented by Dr. David Burden. If you have any questions throughout the presentation, you can type them in the Q&A box located on the right of your screen. If you have any trouble seeing this presentation, we have found that changing your browser helps. Questions will be addressed at the end of the presentation. If Dr. Burden does not get to your questions, my colleagues and I are more than willing to answer them on LinkedIn after the presentation. Okay, without further ado, introducing Dr.
Burden. Hello, everyone. Sorry about that startup. We're a couple technological kinks here. Let me just hide. No, 1 second. 1 minute. Okay, sorry about that. Today we're going to be talking about microbial freeze drying, obviously, and I'll be taking you through that in just 1 second. Just a couple things to start with as to why you should listen to what I have to say. Let me just pull in a slide here. My background in this area is I guess, pretty extensive. I'm a microbiologist, going back quite a few years, actually. All right, so there I am in 1976 when I started working in the lab at the age of 19. So I've been doing microbiology for quite a long time. That's actually up on the island of Nantucket at the UMass Field Station, where I was doing water pollution research and working with some nitrogen fixation in blue-green algae and some other stuff. In this particular slide, I think I'm out trying to catch field mice to isolate Babesia for some people at the, I guess it was Harvard Public Health. But over the years, I've done a lot of different things in microbiology, and freeze drying just one of them. Back in graduate school when I was working with yeast, I used to receive cultures from a number of institutions around the country, and they'd always come in freeze-dried, so I never really did it myself, but I always used to get the samples and have to work with them. And that was my first exposure to it, and that's actually helped me later on when we started developing some of our products for that. So, you know, I've been doing a lot of different things, and over the past, I would say 15 years, fair amount of lyophilization. Okay, quick timeline. So in the 80s, I got my oops, sorry about that. In the 80s, I got my PhD at Rutgers UMDNJ, really working with yeast and genetically engineering it, starting pretty much around 84 when genetic engineering was still pretty young. At that time, I also initiated a bunch of recombinant DNA training courses at Rutgers, which I carried into the 1990s when I set up a training center for doing just that. We're located in New Jersey, and for those of you who've had the fortune to visit this area, it's kind of a lot of pharmaceutical companies right here. So a lot of people needed to know how to clone, and that's essentially what we did, as well as teach other things. And in that, we ended up doing some consulting and contract work, and that led finally into the 2000s, where I established a company called OPS Diagnostics, which took ideas that we developed and turned them into products. And that's really where we are now. We've been focusing on a lot of our products in sample preparation and preservation since that time. So OPS Diagnostics, which is really the host of today, has two major areas that it works in, which is homogenization and also in sample preservation. And in sample preservation, there's freezing and freeze drying. One of my companies I did a lot of work for in the 90s was Revco, and with a lot of cryogenic freezing in their ultra low freezers. And then later I did some work with LabConco, Invertis. So I was pretty well versed on the equipment side, and then we started to develop the materials that go in the machines. So we sell vials and bungs and seals, but our real materials which are novel are microbial freeze drying buffer, which is something that which we'll talk about today, lyophilization reagents, so these are pre-ready, pre-made stuff, and then excipients. So even though we use this stuff, that's really not what we're focusing on. We're going to just talk about the whole process in general. So let me get rid of this slide. Okay, so here we go. All right, a couple of quick bits of information. There's a link. First off, this webinar will be recorded, and we'll put it on, I guess, YouTube, so that people who missed this will be able to look at it later. And so we'll email everybody a link later on. So the notes for today you can find, I think you can find on this site, but also if not on our website at that address, and I believe that's a hot link, which you can click on the PDF, here or on the PDF. We're also going to have a LinkedIn discussion group for those people interested. There quite a few people signed up for today's talk, so which is going to be very difficult to answer all these questions because people have lots of very specific questions. So what we have is a LinkedIn group, and I think everybody's already registered. We just did it by email, so you can click on that, and we'll be in there to talk today and have an ongoing discussion as well for anybody who'd like to talk in this area. Some contact people, the lab people here. We have Rana, Aisha, and Jackie who introduced me, and their emails are there, and you're welcome to contact them. You can contact me as well. However, I have 2,500 emails in my inbox, and sometimes I don't always get to things very quickly, but these people do. So just to kind of keep that in mind, that there's a way to get to us. The 1 second here, just to show you. Okay, there's your supporting cast. That's Aisha, and that's Rana, and that's Jackie. So they're nice, smiling people. So if you have any questions, you can ask them, and if they don't have the answer, they'll relay it on to me. Okay, so freeze drying has been around for quite a number of years. It's over 100 years old. Some of the earlier works were 1910, that area. It really started to develop as a technique in the 1920s when the BCG vaccine came out, and that was a vaccine that was used against tuberculosis. So when that came on the scene, there was a great need to really kind of stabilize samples and make them so you can transport them and everything, and that's when a lot of effort started going into the freeze drying. There was a real lack of standardized equipment. A lot of people, instead of using cold traps, which I'll talk about, used to use desiccants in order to catch water. So they put a vacuum on your sample, and I'll talk about this in a second, and then catch the water with a desiccant, which is not that necessarily that effective. But it was in the 40s and the 50s that people really started to systematically look at the way freeze drying worked and come up with instruments or processes that could be then commercialized. And that's when you really started to see a lot of culture collections being developed and a real progression of the whole technique. So what I'm basically saying is that I didn't invent any of this stuff, but I do use it. And it took a long time to develop. And just as a kind of comparison, at the end of the slides, there's a reference page which with a couple of papers that go all the way back to the 40s. And those PDFs exist. And after you see what I talk about today, you might find it interesting to look at that and see exactly how things have changed. It's always nice to kind of get a historical perspective on things, and this is just one way to do that. Okay, just another quick point is you have freezing versus freeze drying. In both cases, what we're really trying to do is limit the negative effects of water. So when you have water present in a biological sample, all kinds of things can go wrong. You can have enzymatic degradation. You can have free radicals forming. That can just destroy your sample. So in the case of freezing, you bind up water and ice. In the case of freeze drying, you remove the water. And it takes a lot less energy to freeze something, but over the long term, it requires a lot more energy to keep it cold. A quick example of this is that I have a freezer that goes down to minus 140 centigrade. So I have liquid nitrogen tanks, and I have a freezer, and I always liked the freezer because I didn't have to refill it. And in our last location where our lab was, our electricity was paid for by the landlord. And when we moved into our new location about 6 years ago, I found out it cost me about $300 a month just to run the freezer. So once you freeze dry, you don't have to worry about that. You can keep your samples at like in a refrigerator. It's much more cost effective to store than using some very low temperature vehicle with our liquid nitrogen or a cryogenic freezer. Also, for clarification, I'll be talking about freeze drying. It is really the same as lyophilization, but lyophilization can often be used to talk about other solvents, while freeze drying really focuses on water. Okay, so quick aspect of the components of a system. Now, for those who are not familiar with anything about freeze drying, very simply it involves taking a sample that's in water, you freeze it, you put it under a vacuum, and water will pop off go from solid to gas in a process known as sublimation. So that's kind of chemistry 101. In that, you need to have some place where the water can accumulate, which is usually what we call a cold trap. And so the basic components of a freeze dryer has to be over here. You can see a pump, all right, and then you need a cold trap, which is what this center area right there is. And then you need to be able to hook your samples up, which are usually in glass tubes or something, which are on these ports. That's a vacuum gauge, a nice thing to have in a freeze dryer system as well. So the simplest freeze dryer would be what you see right here. It would be some kind of what we call a manifold with an internal cold trap where you would put like dry ice or ethanol to get a nice cold temperature. And this would be hooked by a nice thick vacuum tube up to a pump, and the pump would suck out all the air or all the gas, creating a vacuum, which has to be a very high efficiency, very high level vacuum. And so the pump is really probably the most important part of this system. So you need a pump, you need a gauge, you need tubing, you need a cold trap, and then it gets fancy. You can have manifolds like this over here. This is a lab Conco system, so you have these ports, and you can connect, frozen samples in glass tubes to these ports, and then this is actually a cold condenser. So instead of putting something inside this pot, there's a coil in there that chills up, which will trap your water once it leaves your sample, and then your pump. Over here, this is what they call a shelf freezer, where there's actually, you'll see a picture of this later, but there's a shelf in here where you put your vials inside of a chamber, and then you pull the vacuum on a chamber. And this is a little bit more controlled. There's ways to control the temperature of the shelf, control the vacuum. You can raise the temperature, lower the temperature, do all kinds of stuff, and even stopper the vials that you put in here. This is my preferred method, but they're more expensive. Again, just a quick look at the components here. So here's a manifold with, in this case, a pot. And again, you would put something cold in here, dry ice, ethanol, or acetone to create a cold trap. And then there's it's like a double boiler. There's a space around this, which you can see right here, and the pot goes inside. And these manifolds you can turn them on and off, so you can hook vials or ampoules up to this that have samples that have been frozen to draw on that. Down here, you can see this is the inside of the shelf freezer. And here's your shelf where your samples will sit on. And this shelf, you can control the temperature. So you can heat it up, you can cool it down. And then underneath it, there's a coil, and this coil has refrigerant in it. And the refrigerant can get down to minus, I think this one's minus 50, and serve as a cold trap. So this is where the water from your sample will end up. Okay, so the steps in freeze drying. When most people, or you see descriptions in books or whatever, it often talks about freezing and then primary drying and then secondary drying. And that's all true, but there's actually a lot more to it. You have to culture, when you're doing microbes, you have to culture your organisms, then you have to harvest them, you have to freeze them, then you go through a drying phase, which is known as primary drying, which is the point where you pull the water out of the sample while the sample is frozen and trying to keep the sample cold, but warmer than the cold trap. Then secondary drying is after you remove the bulk of the water from your sample. There might be some residual water, and in something like a shelf dryer, you can pump heat into your sample to push off or basically push out additional water. Then you need to do viability testing. You know, do I have anything alive in my system? And there's usually you can do this through something like a standard plate count, that will show you an easier way to do that today. And then you need to store and you need to monitor your organisms as well. Um, you know, these things there's kind of a perceived notion that when you freeze dry something, it will last for years and years and years, and that can be true, but it's not always true. So you need to really keep an eye on whether these things are alive. And we're going to go over each one of these things in order here. Okay, but the overall objective when we do this is we're going to create a matrix, or what I like to call a cake, that has the water removed. You have you minimize dehydration. One of the things that can happen is any residual salts that are in your sample, as you pull the water out, the salts can start to crystallize and you can pull water into the salts, and you can get localized membrane breakage or something due to high salt concentrations, localized high salt concentrations. You want to, along with dehydration, you want to maintain the integrity of your membranes. You want to retain the 3D conformation of your biomolecules, and hopefully those two things together will allow you to maintain the viability of your organism. So some things to consider when setting up to do this is obviously there's sterility concerns. Um, and there's different views on this. You know, if I'm setting up a bunch of vials that are just going to be stock cultures, which I'm going to use down the road for whatever, whether it's a yeast or an E. coli, I would be probably less concerned about sterility than if I'm making, let's say, vials or ampoules, which would be used as a starter culture for some kind of a fermentation process. The reason I say that is that just like a culture collection, you're going to have 1 million organisms per mil or even more in that little vial. You're going to rehydrate it. In other words, it's going to be a solid material after freeze drying. You're going to add some water to it. It's going to go into solution. You're going to take a loopful out, and you're going to streak it. If there's 1 or 2 microbes in that sample that somehow made their way in there, they're going to be, you know, outcompeted hopefully by the other bugs that survive. And I'm not, you know, please note that I'm stressing that you should follow aseptic technique and everything. It's just that there's different levels of reality, especially when you're using a shelf dryer. But this is something to consider. Do you need to do your stuff at a bench top, or do you need a clean room? It's just things to think about. The other thing is the format. Are you going to use vials, which are glass vials that are stoppered with bungs, or are you going to use ampoules, which are sealed under a flame? You can also do bulk freeze drying in a bell jar. I'm not going to talk about that today, but that's just kind of a bulk method. You need to know what kind of solution or buffer you're going to freeze dry your stuff in. What I call them, just generally calling lyophilization reagents, have to have a lyoprotectant that protects your organism, but ideally it will also have something to help form a nice cake or solid matrix for your organisms to be suspended in. Now the ATCC has a real nice reagent, something called reagent 18, which is a pretty straightforward reagent that consists of, I think it's about 10% sucrose and 5% BSA and a little bit of tryptic soy broth. And reagent 18 is a very standard reagent and it works very well. We have a product called Microbial freeze drying buffer, which is essentially the same thing, but what we did is we took the bovine serum albumin out and replaced it with a plant protein, and we also took the tryptic soy broth out. So it's very similar to reagent 18, but there's no animal products in it. And in reality, a little plug for our company, the cost of BSA is so high that we can sell a 500 ml bottle of our stuff for less the cost than the BSA is used to make this equal volume if you made it yourself. So it's a win-win, actually. It works very, very well, and I'll show you some stability data on that later. Other additives that are often used, lyoprotectants, sucrose and trehalose. These are both disaccharides, and they have the key factor of that the anomeric carbons in these are blocked or actually linked to something. So in sucrose, which is a glucose linked to a fructose, the anomeric carbon, the alpha beta carbon, for those who can't remember what anomeric is, is linked to the fructose. Trehalose is a alpha 1,1, I believe, glucose molecule, and for reasons which I don't know if anybody knows the reason, these anomeric disaccharides or non-anomeric disaccharides are very, very good for protecting molecules during freeze drying. Caking agents such as BSA and skim milk are good for making a matrix, a 3D structure, which allows you to freeze dry. And that 3D structure is very important because water needs to get out of the structure. And if you don't have enough structure, your sample will, what we call, collapse. And in collapsing, you end up drying your sample as opposed to freeze drying your sample. And then the last thing to consider is your sealing. And in a shelf freezer, you would seal with a bung. A bung is just a stopper, but it has a little split in it so you can put it into the vial and gases can get out. The alternative to that is the flame seal, which is what you do with an ampule. And in flame sealing, I'm holding my hand up right now. I've never flamed an ampule that didn't look like a mutant. I'm just not that good at it. But you buy like some kind of a standard, and it always comes in a nice flame sealed ampule. I'm not good at that. But if you are flame sealing, you'll need some kind of a torch, an acetylene or oxyacetylene, propane, butane. The hotter the better. Acetylene torches are nice and hot, and they'll seal very nicely. But there's a lot of tricks to flame sealing, which I'll discuss a little bit later. Okay, so the vials. Vials are glass. First off, everything you freeze dry in should be glass, and I'll tell you some don'ts in a second. But these are glass. You can get them different volumes: 2 mil, 3 mil, 5 mil, 10 mil. We sell, I think, 2, 5, and 10, I believe. But here's the bung right up. Let me get my arrow. That's the bung over here as well. And there's a little gap in that bung, right? See it right there. And you put this in, and the gases can get out. So your sample is placed in the bottom. You freeze it, and then you put the bung in there, or you put the bung in, then you freeze it, put it in the dryer, and pull the vacuum, and your gases can get out. After it's done, this is pressed down to seal it, and then this little foil seal is put on top, and you crimp it on. So vials are like if you have a shelf freezer, they're very commonly used. You can get them from a lot of different places. The one thing is when you use vials in, let's say, a shelf dryer, they got to be bunched together. So you cluster them together. Like our freeze dryer holds about 280 of these things, so they're all packed together, and that helps in the heat transfer. You need good heat transfer in order to get the water out of your sample. Ampoules, just a selection stolen from the internet here. Again, they're also glass. They have long necks, so you put your samples down here, and normally you would put like a piece of glass wool or something in here in order to seal this off so that when you're handling it, you don't have to worry about contaminants getting back into your sample. One of the things you have to worry about with these, if you've ever worked with ampoules, is that when you load these up, you're going to need a long neck pasture pipette. So if you have a culture and you want a pipette in the bottom, a micro pipette won't work because you'll end up getting all your liquid on the neck, and then that just makes for a messy situation. So your sample is placed in here. You put some cotton, or it's not cotton, that will burn. You put glass wool in here that's been sterilized, and then you freeze this. This is put onto a freeze dryer port, and you pull a vacuum and it will dry. And again, there's some tricks to that I'll get into in a minute. And after it dries, you would actually come in with a flame. Let's say right here is your glass wool, and up here it's connected to a manifold. You would then have to flame seal this right here. And the idea is that you hit it with this torch, and the vacuum pulls it in and it closes off. Like I said, it works, but mine always look very ugly. Therefore, I use a shelf dryer. Okay. The other thing I don't like about Ampoules is that to open them, you have to snap them. And I know at least once I've cut myself doing this, which is not necessarily a fun thing. So you have to be careful. Get pre-scored Ampoules. They have a little kind of ridge cut around there so that they snap off nicely. Okay, things you don't do. Though it's tempting, do not freeze dry in microfuge tubes. So plastic is a big no. And I've seen a lot of people who've tried to do this. Plastic is actually an insulator, so heat doesn't get into it well. So if you have a sample and it's frozen in your tube, for heat to kind of diffuse into the sample and for it to push the water off, it doesn't work well in plastic. So microfuge tubes are not a good idea. The other thing is after you seal your microfuge tube, and this I figured this out the hard way, which I can show you some indicator later where you can test this. Water does seep across, like diffuse across plastic over time. So if you're trying to keep your samples nice and dry, plastic microfuge tubes is a bad idea. Same goes for like polystyrene 96 well plates. My original product that I developed for freeze drying was just that, was for 96 well plates. And after I did a whole bunch of work, I discovered that once you freeze dry in a 96 well plate, you can actually have water will diffuse across the plate. One thing you can do is, if you do use plastic, which I don't recommend, is you can't put your samples into like a seal and meal bag, a Mylar bag, and vacuum seal it after you freeze dry, but you have to work quickly because it will pick up water pretty quickly. Don't use any old pump for freeze drying, all right? You need a good pump, which leads me right into the next thing, is I've seen people who've tried to freeze dry using a water aspirator. Another bad idea, considering you're trying to get the water away from this thing and you're using water to create the vacuum. Bell jars stuffed with vials doesn't work well because you get uneven heat transfer. You need to really, if you're going to do massive vials, I would recommend using a shelf dryer. Some people will think that glycerol or saline is a good idea with freeze drying, since glycerol is good for freezing. Glycerol doesn't vaporize, so it will gum up your sample. And saline, such as PBS or whatever, will cause, you'll end up with a lot of salt, and the salt is bad for your sample. So these are things which you don't want to use. All right, so the culturing. We are talking about microorganisms. My preference in doing this, and we've done a lot of work. We do some contract work on freeze drying for people. And cultures are always the best. You grow it up, shake flask ideally, and all you have to do is harvest your cells and dilute them in or resuspend them in one of our buffers, and then aliquot them and freeze dry. Sometimes that doesn't work so well. You can use solid phase, so you can grow cells on an agar plate, and then you can put like reagent 18 or microbial freeze drying buffer on the plate and flush off the cells and just drop the medium and then freeze dry that way. It doesn't really matter what you do, but what you want to do is get rid of the growth medium and replace it with the lyoprotectant. Now, the exception to this is that anybody working with fungi will know that they don't like to cooperate. On the one hand, mycelium, you can end up with a big fungal mass that just doesn't want to dissociate or disaggregate, as I put it here. So what do? You end up with, you have 10 mils of microbial freeze drying buffer and a big blob of fungus in the middle of it, and you're trying to break it up. There's not a good answer for this. If you can break it up without destroying the viability of the cells, then try to do just that, pipette it up and down or whatever the case may be. Or you can just freeze dry the big blob. Spores are a big headache because a lot of spores are hydrophobic and they float. So if you have a plate, let's say you flush it, you'll find a lot of the spores will float to the top. You just got to shake it up the best you can and try to get the cells to disperse. But you can't really add, let's say, like a tween or something like that to help disperse them, because that will end up hurting your overall freeze drying process. The freeze drying medium, historically, glucose and BSA were used. They were replaced by skim milk and sucrose. If you go back to a lot of the culture collections, skim milk is used all over the place. And the thing is that skim milk really isn't that good of a protectant. It's a really good matrix. It forms a really good cake, but it doesn't protect your cells very well. So what you really want is a combination like sucrose and BSA, which is what's in the ATCC solution reagent 18, and that's similar to what we use. Like I said, reagent 18 has sucrose, BSA, and tryptic soy broth. Our version is essentially the same, but we take out the animal protein. So, but you want a medium that really has both the protectant and the caking agent to give you the best of both worlds. Some people will talk about using like mannitol, because that's used in, let's say, a lot of drugs, or trehalose, and I've used those, but I don't necessarily find them better, or there's sometimes they work less well. So I kind of stick with what I know. Okay, so you harvest your cells, you spin them down and resuspend them in freeze drying buffer, or you can flush your cells from a plate, but then you dispense it. So you need to either go into vials or ampoules, you plug them, and then you start the process. I would recommend, if you're doing a lot, if let's say you're working on a culture collection, get your hands on a repeating pipette. You have the kind of the big syringe type. They're very, very good for doing multiple samples. We did a project, I think, last summer where we did almost 400 different strains of a bacteria, and we would have gone nuts if we were trying to do that with, let's say, a regular pipette or even a micropipette. So some tools, that's the kind of thing that's really helpful. As I mentioned, with ampoules for filling them, sterile pasture pipettes are critical for getting the solution way down into the bottom of the ampule. Sterile glass wool in the neck of the ampoules is recommended. And as I said, for sterility concerns, for especially shelf dryers, a clean room is oftentimes warranted. We have a whole bank of hoods, what are known as aseptic filling hoods, and those can be used pretty nicely as well. You can move the freeze dryer right up to them if you need to. So the freezing, there's two ways to freeze. Either you can do it by hand, or in let's say a dry ice ethanol bath for, let's say, an ampule. So over here on the images here, we have something called a cryo cooler, which is a device we make for actually storing things at liquid nitrogen temperatures. And there's a well right here, and in this well we put liquid nitrogen. And what I'm about to tell you don't do, by the way. I have a little example of how things work. So I wanted to freeze my sample, and in this case I was using both vials and ampoules. So I would take them and I put a glove on and I inserted them into the liquid nitrogen, and I can do one of two things. I can just put them down there and I can freeze them straight out, or I can put them down there and I can twirl them. And in twirling them, you basically end up coating the sides of the ampule and the vials with the buffer and your cells. Now that's important because of surface area, which I'll talk about in a second. But in the freezing, you have some choices. All right, you can use a shelf freezer like I showed in the shelf dryer, and that can go down to like minus 40 or so. Good temperature. You put your samples in there. Temperature drops over time. You can program it, and your samples will freeze. Or you can use a lab or a minus 80 freezer. You can put your samples in there just in a tray and let them freeze. Or you can use something like a dry ice ethanol bath, where you put your samples in it and freeze it. Or you can, like I said, you can twirl them and freeze them. In any case, whether you're using if you're using like a lab or a minus 80, you can also put your samples in, or your vials, and put them on a tilt so you can create a slant. And that idea of freezing on a slant or freezing by twirling the vial in the ice bath is important because of surface area. You want the maximum surface area to the volume of the material so that you can get the water out quickly and it doesn't have to go a far distance. If the water has to travel a far distance to get out of the ice, then you have a good chance that it will warm up and your whole sample will melt before it's dry. So that's something, I'll show you some images on that in a second. Some considerations in freezing temperatures. The freezing temperature should be higher than the condenser, and the condenser meaning the cold trap. Now when I say it should be higher, keep in mind that minus 20 is higher than minus 80. So what I mean is that you don't want to take, and which I'll show you in 1 second, samples and put them in the liquid nitrogen to freeze them, and then hook them up to a freeze dryer that has a condenser at, let's say, minus 50, because the water will flow from the warmer temperatures to the colder temperatures. So in that instance, your samples are actually acting as the cold trap, and you don't want that. You want to go from you want your water to go from your sample to something colder. The other thing to consider is the rate at how fast you freeze. So you have slow and fast freezing, or in other words, like flash freezing. When you slow freeze, your ice crystals tend to form and be larger. So you end up with larger ice crystals and basically more channels for water to get out of your sample. If you flash freeze, you end up with many tiny ice, nucleation all over the sample and many tiny ice crystals, and that's better for the cells and the biomolecules, but it's harder to get water out of. So just something, you need a balance between something that's really good for your sample and something that water can get out of easily. And normally when you're dealing with something like a bacteria, the efficiency is normally so high anyway that just freezing at, let's say, minus 20 in a regular freezer will usually work well enough to give you good viability in the long run. So sample freezing. So shelf freeze dryers, you can program it. For instance, I will take and I'll put my couple hundred vials on our shelf freezer, and it will start at 4 degrees, and I'll set it to go to minus 40 over, let's say, a 1 to 2 hour period. Hopefully, at the end of that, if all the vials are flat on that surface, and you give it a chance to really chill down, everything will be ice. Occasionally, if one of the vials is not touching well, it will still be liquid, but that's only 1 or 2 out of the entire run if you're not careful. Flash freezing, as I noted, increases the number of ice crystals so that they're more numerous and they're smaller. It's better for your sample, but it's harder to get the water out. Slow freezing is pretty much the opposite of that. You may have more extracellular ice, which may cause water to diffuse out of your sample. You might get some dehydration, but overall, to get the water out when you put the vacuum on, it becomes easier. So you got to balance here between these two. All right. Normally, like I said, with your average bacteria, a slow freeze will give you a better result. Any way you look at it, you have to maximize your surface area, especially if you're using a manifold or like an ampule. You really have to increase the surface of your area to as much as possible. All right, some important points. Maximize the surface area. These are just some examples of how I did this. Here we have a vial, which I literally put on its side when I froze it. So there's 1 ml of freeze drying buffer in there, and you can see it's a huge, basically surface to volume area, or ratio if you want to call it. This is an ampule. You can't see this. I took a bad picture. I apologize. But on the back side of this, it's at an angle. So this one is also I'm trying to maximize that surface area. So in ampules, and these are both hooked up on ports, by the way. When you're doing freeze drying with a port, you want to really keep your volume small. You want to freeze on a slant, and ideally freeze at minus 20, so that you get it's nice and easy for the water to get out of the sample. Now, I talked a little bit ago about what not to do. Here are a bunch of samples that I freeze dried using liquid nitrogen, and I want to show you the result of these. So up here we have four, I think these are 10 ml vials, and I have these stoppers that you see right here that you can push in them, and then this will hook right into the manifold on the freeze dryer. So this has like 5 mils, this has 2 mils, this has 1 mil, 1 mil. So these two I just froze straight off. They kind of look like that, all right? And then I took one and I just hooked it up without freezing it at all. And then I took one and I froze it just flat and I hooked that up. So I hook it up and I turn it on. So vacuum's on all of these things. And what you get is that initially when you turn it on and you don't freeze your sample, some people will suggest, well, you just put it on there and turn it on and you'll pull a vacuum and it will freeze your sample right off. And that's not necessarily true. What happens is you'll see a lot of bubbling and then it will calm down and you'll end up with liquid in there, and over time that will dry into a flat crust on the surface of your vial, and that's dried. It's not freeze dried, that's just dried. Down here, since there's such a distance for the water to go through, this will start off and look fine, but over time all of a sudden it will melt down and it'll turn into a liquid, what we call melt back. And in this particular case, water is being drawn off. It's just going from liquid to gas, and you actually end up with like a skin forming due to just proteins and stuff accumulating as the water is being removed. Here, this looks good. You can see it's frosting over, but since it was frozen using liquid nitrogen, a lot of little ice crystals. It's flat, so it started going pretty well, and then towards the end it started to melt. All right, and what happened is initially some melted underneath in the bottom, and when you got the liquid there, it formed gas, and it actually pushed the dried cake up this a little bit, and then some of the water on the top melted, and when that happened, that bubbled up and formed a film. So this is kind of like halfway between good and bad. There is a cake in here, right? But it did melt in the process of being freeze dried. So not the best result, but definitely better than the other two. All right, so if you freeze using a warmer temperature, and I mean not warm, but minus 20 to minus 40, what's the result? So here's the sample I had before that I showed you in the Ampule. And this was placed on the freeze dryer, and after a couple of hours, this is from a better angle, this thing is completely dried. So this is about 250 microliters of sample after about 2 hours, and the sample is dry. And this would all I'd have to do, I could seal it at this point and take it off. Over here, I had, just like I did with the liquid nitrogen, I had a sample where I froze flat in a vial, put it on the freeze dryer, and by the time it was done, it was liquid. All right, so that melted back. Here's the same sample, same volume anyway, which I put it on an angle, and I let that go, and then it ended up drying. It's a little bit hard to see, but the edges are all kind of opaque, and it dried relatively well. So just the fact that this had a greater surface area, and it was frozen at a higher temperature, which allowed for greater pores for water to get out of, the result was much better. So the bottom line is that if you're freeze drying using a manifold, keep your sample small and try to maximize your surface area. And don't freeze at a super cold temperature. So in the primary drying, once you freeze, if you're using a shelf freezer, once it's frozen, you just turn on the vacuum. If you're using a manifold, you push your sample into the manifold and turn the valve. You need to reach a vacuum that's below 100 millitorrs, which is all these things over here. So you're talking about 10,000th of an atmosphere, very, very high efficiency. And once you're there, you have to let it go for, depending on your sample, usually at a minimum overnight. All right. So especially if you have a lot of samples hooked up to one freeze dryer. So the time is going to be dependent on the volume and the sample number, but overnight is not uncommon. If you have a large volume and let's say a shelf dryer, you can go several days. So here we have the samples I just showed you a second ago. Here are some samples that were in the shelf dryer. Actually, I'm looking through the door so it's a little shiny, but I have a indicator reagent here, something we sell called freeze drying indicator. And this actually shows you that you can see a little kind of white rim here and a little light rim there. And that's the beginning of the freeze drying. And then over time, it will end up looking like this. Let me pull in another slide here, another image here quickly. I actually did this last night, and whoop, there are the girls again. We call them the lab rats, by the way. Okay, here are the picture of the indicator, and this is what it looks like after. This is overnight. So this is taken out and stoppered with a bung. So this cake is nice and dry, and this is the advantage of a shelf dryer, in that the shelf was maintained at minus 10 degrees, so that the sample never really had a chance to melt. But you can try to get a big differential between the condenser, which was at minus 50, and the sample, which is at minus 10. And you can control that in a shelf dryer. So this dried very nicely overnight. Next to it, where'd my little arrow go? Oh, I'll get another one here. Okay, so this is overnight, and that's microbial freeze drying buffer. And this is our indicator, and you can see that there's a little residual water on the surface, but this is blue, and that blue is like 1 to 2% water. So we use this to kind of map map freeze dryers, and also to let us show when some processes are done. All right, let me get that out of here. Okay, so let's continue here. Okay, the secondary drying is not really practical when you're using a manifold, except to say that it happens naturally. In other words, your sample starts cold as you take it from the freezer or from a bath. You hook it up, heat the vacuum pulls heat out of the sample, heat diffuses into it, and it continues to be cold until the water's gone. And then that's going to be essentially your secondary drying. So your heat goes in just from ambient, and water's pushed off. If you have a shelf dryer, you can control that temperature. So where I would, let's say, keep the samples at minus 10 during primary drying, at secondary drying, I might bring it up to 20, 25 degrees for a couple of hours. And this is, again, the aim is to drive off remaining water. The secondary drying phase only lasts a couple of hours, and after that I go on to sealing. All right, so sealing, you do ampules with a flame. You can't seal the you can remove the ampule from a manifold and seal it, but keep in mind that when you take a sample off of a freeze dryer, if it's open to the air, it will pick up water very, very quickly. There are automated ampule sealers, but if you ever look at them, they're on a little carousel, and I mean that's not a practical way to handle something if you're going to be trying to freeze dry and keep it viable. That's good for liquids and ampules and that type of thing. So usually try to seal it while it's on the manifold with the vacuum. Vials you can seal with a bung under vacuum. So down here in the bottom is the shelf dryer, and there's a stop ring mechanism that's hooked up to a piston. So in this shelf there would be your vials, the bungs would be in there, and I simply hook it up to a pressure tank and push this panel down, this stop ring mechanism down, and push the stoppers into the vials, and then I can release the vacuum. After your sample, whether you do a vial or an ampule, people even use vials with little screw caps, for instance. You can throw them into a Mylar bag with a little silica desiccant and a seal-a-meal bag, and seal them that way if you're concerned that your mechanism for sealing is not that accurate. Those systems can seal out the water very effectively. Storage. Temperature of storage is a big deal. Cells will not remain viable forever, and if you try to store your cells at 4 degrees, you have your cells should remain viable. Excuse me, But if you try to store them at a higher temperature, you might have some real issues. Um, we, in the past, have had people who have insisted that we do accelerated shelf life on freeze-dried cells, and it's a real disaster. I'll show you some data in a second, but where a cell stored at 4 degrees could last for years, something stored at 37 may last only a few days. And I think it's because there's so many different components in a biological system that can go bad, as opposed to, let's say, a solution where there's, let's say, one protein or something, that the breakdown of the sample, or the drop in viability is extremely pronounced. But however you store these things, it's important to test them. And I would start off by testing a sample, let's say once a month, and then after I see it's not changing, once a year, and then every few years. And it will just let you know where your samples are and whether you need to grow them up again and start your process all over. All right, so here's just a little data from way back when on the viability of cells stored at 37, and this is under 3 different buffers that are being used. So we had sucrose, trehalose, and reagent 18, which is sucrose and BSA. And this is real time over 7 days, and they start off with pretty good viability in a log scale here, and then over just a few days, the trehalose cells are down a whole lot. You know, you're going from 10 to the 8th to basically 10 to the 4th, and the sucrose, you're down a couple logs, and even that's the reagent 18, you're down a couple logs, and the sucrose, you're down about just a log. So that's over 7 days. So you can imagine that if you let this go for a year, things wouldn't look very good at the end of that time. So in discussing that, how do we do our viability testing so you can test this? And I'll get back to that that stability in 1 second. But traditionally, people would do plate counts, just standard microbiology, with serial dilutions and pouring them out in plates and counting them. I find that a tremendous amount of work. I find it uses up a lot of supplies, uses a lot of incubator space. So we have a simplified approach, which is, in my opinion, good enough for what we're trying to see, which is essentially a relative idea of how viable things are. So we use a idea of dilution to extinction, which is it's easy, fast, cheap, small footprint, and gives an answer that I want to see, which is either I have a lot or a little. And it's quite simple, really. This comes back from anybody who knows old microbiology of something called most probable number. That's kind of where I got the idea from, which was back what we used to use in the 70s for counting microbes from water pollution. But in this, all we do is we take a plate like this, a sterile plate, put a broth in it, so it's tryptic soy broth or whatever, and we take our vial. And one thing I don't have down here, which I meant to put in, is after you freeze dry your sample, you have to use it, rehydrate it, and there's a lot of different ways to do this. I just take my sample, and since there's already a broth or protein in there, I just add water. So if I freeze dried something in 100 mils, in a mill, I'll just add a mill of sterile water to it. I'll let it sit for a few minutes to give the cells a chance to wake up, and then I'll use it. So when you take your sample or your freeze-dried culture and you rehydrate it, to do this technique, all you would do is put in 1/10 volume here of something. So I used a multi-channel pipette and put in, I believe, 180 microliters of broth in all the wells. Came along with 20 microliters of sample in the wells, and then proceeded to dilute by transferring 20 microliters to the next well, changing my pipette tips as I go. So this is basically each well represents a log of dilution. So in this case, I diluted out, and when I got to 10 to the 9th, nothing was growing, and 10 to the 8 something was growing. So what I say is that I had 10 to the 8 cells to simplify it, and that's how I count them. So when I viability test to get an idea of efficiency, when I prepare my cells, I'll use half the plate and do this so I can see what the cells look like before they get freeze dried. So let's say it comes out to 10 to the 8, and then I freeze dry, I take a vial, I rehydrate it, and I do this, and hopefully I'll get the cells will grow to the same extent, which case I say I have 100% viability. I consider myself successful if I can get to within 1 log of the pre-freeze-dried sample. So if I get to 10 to the 7th, that's not great, but it's still pretty good. You're talking about billions of cells. If I'm way down here at 10 to the 3, I have a bug that's just not cooperating, and I can either be happy with that or I can try to do something else and try to make it more viable. Some organisms just don't like to cooperate, and that's just life, but you have to try to work around it. So here's some real-time stability data. So this is E. coli we did in 2010, and we started off and they all looked nice on our dilutions up here. It was between 7 and 8 dilutions out, they were fine. And so we looked at skim milk and sucrose and microbial freeze-drying buffer, and our original formulation had tryptic soy broth, but that TSB has a little bit of animal protein in it, so we did away with that. And now we just use microbial freeze-drying buffer without the TSB. But over 70 months, the skim milk ended up coming down to nothing, all right? And this nothing was just actually last week for yucks. We pulled our samples out of the refrigerator where we keep them at 4 degrees, and the skim milk was dead. The sucrose was down to 10 to the 3, so that dropped basically, I don't know, somewhere between 5 logs, maybe 4 to 5 logs, so significant drop off. The microbial freeze-drying buffer dropped a couple logs after 6 years, and then the buffer with the TSB did better. So even here, even though our current product is a little bit lower than the one with the animal protein, we kept it, I'd still consider that good because this could go on for another 4 or 5 years before we'd have to reculture it. So this is real time, all right, but it gives you an idea that what you freeze dry in will make a big difference in the long run. Okay, so how's this going to work on the stuff you're working on, which is always what people ask me. I get emails all the time. I'm working with some, esoteric organism. Can I freeze dry it? Yeah, you can freeze dry it, but I'm not sure if it will be alive. And the bottom line is that every strain is different, and that's a horrible answer, but it's a true answer. So, if you need to freeze dry something, will it work? And the answer is probably. Most of the stuff that we freeze dries without a hitch. But bottom line is you got to test it. And the same thing goes with how long will it last, and it'll probably last a long time, but again, you got to test it. And will it be efficient? Probably, but once again, you got to test it. And as simple as I can put this is that most of the microbes are easy to handle. They're not a big headache, but there's always going to be exceptions. As I mentioned, we did that big project last year, and hundreds of strains, and there were a handful that were just nuisances, but there's just no way around it. Some are like that, and it's something to do with the biochemistry of that particular strain. It was all the same species, just different strains. As you're going out there, always keep in mind that you need to look at your bugs and test it, and if there's a headache, you got to maybe tweak your growth medium or tweak what you're using to do the freeze drying with in order to get good results. Okay, a couple of links for you here. We have a bunch of pages on our web site that deal with freeze drying or preservation. It's actually these pages, we watch the numbers of people who come in and look at these things. And through these few web pages alone, we have over 5,000 visitors a month. that come in and look at these papers or ask questions based on these papers. So that's why we decided to put on actually a live talk. So, but these are here. Feel free to look at them, and they're hopefully they're straightforward in their explanation. So that's there, and then also just a couple references. These manuals up here are kind of dated, but they're like I said, freeze drying's been around for a long time, and these are just simple how-tos on how to set things up, and they'll talk more specifics on, let's say you're working with a yeast or fungi or bacteria. And then again, I noted that we had some early research papers that that are just interesting to kind of get a comparison about what we do now versus what was done in the development of this whole technology. So with that, I believe I'm done. I am, and I think I'm being handed here a note. These are some questions, which I will try to answer at this time. All right, so the first question I have is, can you freeze dry in a shelf dryer multiple strains of a bacteria without cross-contaminating? And in true aseptic technique, I would say no. In practice, you probably could without too much of an issue. One of the things that you might want to do in such a situation is, if you can contain your organisms, let's say you put them into bags, let's say fill them and let's say put them in plastic bags and freeze them before you ever put them in the shelf dryer, then there's not going to be a lot of aerosols because you froze everything. So you can actually migrate them to different sections of the dryer, and then once they're frozen, you're going to stop. Once they're freeze dried, you're going to stopper them. So you may want to first freeze them outside the dryer and then put them in the dryer. Okay, next question we have here is, how will a strain maintain its viability? Oh, that's a question. I don't know if I can really answer that. Essentially what the freeze drying does, based on the lyoprotectant, is the lyoprotectant, and why a disaccharide with no free anomeric carbon, I don't know. But basically what the disaccharide has been found to do is to fit in to biomolecules, very specifically proteins is where they're best known, and it takes the place of water, so that when the water is pulled out, the lyoprotectant prevents the denaturation of proteins and membranes by, like I said, serving to replace the water. So in the freeze drying process, what you're really doing is first, freezing the sample in the presence of a lyoprotectant, and then removing the water, and the lyoprotectant takes the place of the water. That's why you saw on that one slide on the freeze drying using skim milk, is that over time the skim milk really, the viability of the skim milk really, decreased rapidly because there was no lyoprotectant in there. But if you had the lyoprotectant, viability lasted for a longer time. Oh, I have a question here. Can my company ask you to freeze dry for us? Sure. We do that. This is not a plug. This is a Russian, as far as I know. And we can discuss any of that. Is it minus 20? Why not minus 80? Well, you can freeze at minus 80. As long as you realize that at minus 80, you're, you might end up with like smaller ice crystals, the sample. What? Just told to slow down. You can't hear me. At minus 80, the sample is, you can freeze your tubes at minus 80, but you might want to first insulate them so that the slowing process is it's just slower, much like you would do with a cell line that you're using a minus 80 to first reduce it in temperature prior to the actual freezing. Let's say in a liquid nitrogen tank. So if you put it in, let's say a cooler and put it in a minus 80, the rate of freezing will be slowed, so it will still freeze, but it won't flash freeze. So minus 80 can be used. it's just at a -20, you'll definitely end up with bigger, basically pores for the water to get out. Next question was, how important is the ratio of lyoprotectant to cells? You know what? I haven't looked at that, and what I can tell you is that when I take a culture and grow up, I usually resuspend the culture in the same density as I took it out of the shaker or wherever I'm growing the cells. So if I have a 10 mils of a bacillus that I'm growing and it's a murky culture, I take it out, I spin it down, and I resuspend it. Now in doing that, my objective normally is just to preserve cells for like a culture collection. So I'm not trying to develop a process or anything. You may want to, certainly if you have a very dilute culture, you could spin it down and bring them up in, let's say, 1/10 the volume. You certainly can concentrate your cells. Where you might have a problem is if you try to freeze dry a paste. I think that could be a real issue where you may not have enough lyoprotectant for the number of cells in there. But I know that just going from a standard culture concentration to the same concentration using the lyoprotectant, you're usually in pretty good shape. And let's see, next question. That's one question? Okay. I read that in your microbial freeze-drying buffer, you suggest equal volume. How feasible would it be if which is that, one what? Oh, if I am to grow cells in 6-liter volume. Ooh. I'm not sure the question, or, I mean, you can certainly grow your cells in that amount, and you can certainly resuspend it in that amount. If your cells are very dilute, then you might want to concentrate them. Or if you're going to do 6 liters, I mean, you can certainly use a larger volume of material, but you're going to have to, the freeze drying is, you're going to have to dispense it in order to do that, unless you have some kind of a process scale freeze dryer, which certainly exists. And one last question. Can you use Falcon tubes in freeze drying? Falcon tubes are plastic. I would not recommend it because ultimately when you seal off your tube, moisture can get through the plastic and seep into your sample. I know we're all kind of taught that plastic is impervious to water, but only some plastics are impervious to water, not all plastics. So I would not recommend using Falcon tubes. All right. With that, I would like to thank everybody for listening in today. We will have our LinkedIn site, and feel free to go in there, and we'll be checking that. Maybe not immediately. I have some other things on my plate today, but we'll be going in and answering questions, and feel free to come back, and we can keep a discussion going for as long as you would like. So again, thank you very much, and I hope this has been very useful. We'll be sending a link with the recording once we get that up, and therefore you can visit this again if necessary. So have a good day, and by the way, we're going to be having another webinar in about 4 weeks, I think, and this one's going to be on high throughput processing. I believe some of you may be signed up for that. Using basically bead beating in a high throughput format for if you have to process thousands of samples, how do you go about doing that? So, okay, thank you very much, and have a good day.